Before we get started, just two quick reminders. First of all, GSA online events are governed by a code of conduct, which is now available at the link in the chat. Second, if you have questions for the speaker, please enter them in the chat window and we'll do our best to get to as many as possible after the talk. So today we're honoring the 2021 recipient of the GSA Medal, Douglas Koshland, for contributions to the field of genetics in the last 15 years. Doug is a professor of molecular and cell biology at UC Berkeley, and the medal recognizes his contributions to our understandings of chromosomes and genome stability. Through creative technology development and experimental ingenuity using baker's yeast, his work has illuminated our understanding of chromosome structure, how damaged DNA is repaired, and how cells are able to tolerate stressful conditions. He and his colleagues co-discovered the SMC proteins and then were the first to develop fish assays for chromosome condensation and cohesion in yeast. With this new tool, his lab and others showed that these conserved ATPases mold chromosome structure as part of the cohesin and condensin complexes. He went on to explore many aspects of the structure and function of these complexes, for example, revealing roles for cohesin in both sister chromatid cohesion and double-strand DNA break repair, delineating the pathways for activating these distinct functions, as well as making important insights into the mechanism by which cohesin tethers sister chromatids. The Koshland lab has also made many discoveries in the area of genome stability, particularly in the role of RNA-DNA hybrids, and in the area of stress tolerance, how trehalose and hydrophilins act synergistically to enable organisms to withstand desiccation. Doug's career has been marked by his focus on challenging problems and the creation of foundational methods to address them, methods that have become widely adopted by the rest of the field. As Jasper Ryan put it in his nomination, Doug is a pioneer who opens up the ability to study things that people had not considered approachable. He and his colleagues have made a breathtaking range of discoveries. Doug, on behalf of the GSA Board of Directors, I'm proud to virtually present you with this year's GSA Medal. I hope everyone out there will join me in applause. And now I hand it over to you, Doug.
Okay, well, thank you for the very kind introduction. What I thought I would do, and thank you the GSA for this remarkable honor. What I thought I'd do was begin with a few introductory comments about how I got into genetics, what this award means, and why I love genetics. And then we'll get on to the science. So let's see what we're doing here. Okay. So my journey into genetics. As an undergraduate, I took an introductory biology class, and I hated the genetics component so much that I became a chemistry major and never took another biology class. And in that time, I did an internship with Arthur Kornberg, who was the biochemistry evangelist. So I went off to graduate school at MIT thinking I was going to be a biophysics major, but MIT made me take an undergraduate course in genetics, and there it was taught by a genetics evangelist, David Botstein. So David was a better preacher than Arthur, and I joined David's lab. And then I told Arthur, and Arthur respected David, but genetics not so much. But nonetheless, I started my road to learn genetics, and I've never felt I've mastered it because along my career, both in my graduate work, my postdoctoral mentor, and the many colleagues I've had, I felt that they are all much better geneticists than me, true masters. Well, maybe not Gottschling and Petey's. So here I am with this award, and I want to talk a little bit about what an award like this means. And so I looked through a bunch of speeches, acceptance speeches that I've heard and or seen in my life, and I remembered 3 acceptance speeches done at the Lasker Awards for the cell cycle regulation, very, very short speeches. The first was by Lee Hartwell, who said, "I accept this award for the field." And I think it's a reminder that it takes a community to build any body of knowledge, and that we stand on the shoulders of those before and around us. And for me, very fortunately, I've stood on, in the work I'll talk about today, there have been many, many people who have contributed gems that which we use as the basis for our work. I've also stood on the shoulders of all the people in my lab, past and current. This is every person that's ever been in my lab. And they were amazing scientists who really were responsible for the discoveries. So the second speech was by Paul Nurse, who said, "I'm so honored to get this award, two scientists that I respect so much." And I think, that reminds us that the remarkable collegiality that we have in science, this sort of common spirit of integrity, of joy of discovery, and, in sharing ideas and reagents. And, I've been fortunate, of course, these people that I had in my lab were not only scientists, but they were amazing colleagues, and it's been a really fun ride. But also that I've been blessed with really good colleagues, in part because I joined the yeast community, which has a tremendous standard of sort of collegiality, I think imposed by the leaders who had this standard, really a high bar of generosity and scholarship. And, if you want to make a modern definition of being ostracized, it would be Lynn Cheney and the Republican Party, or a yeast geneticist who won't give out their published strains. So the last speech was by Yoshi Mizui, who said, "I can't believe I'm getting recognized for playing around with egg extract." And I think that reminds us all really how lucky we are that society gives us the financial support to pursue our creative explorations. Since I have siblings who are artists, I just want to point out to you, if you look at the percentage total annual federal budget, 45 billion on biology versus the arts, the National Endowment of Arts gets 0.2%. So, I think we can do some deep soul searching and ask ourselves, is the outcome of the most creative exercise in science that much more beneficial to humanity than creative exercise in the arts? And for those who think the answer is obviously yes, I challenge you to send me a tape of you convincing a friend or a parent why your research will be relevant to their lives to the tune of $450,000 a year. So just to make clear here, in case this goes viral or something, I'm not proposing that we cut NIH. I'm just thinking we should feel lucky, and that hopefully someday we'll fund the National Endowments of Arts similarly. So I want to thank all the funding that I've had out throughout my career, and sort of want to end this segment by just saying that I hope, the real value of this talk will be that you all sit around with a beer and outside and think about how lucky you've had to have the field that you've been involved with, your colleagues, and the fact that you've been supported in your careers. So why I love genetics. So the first four independent important reasons are: one, I'm a chemist in heart, and it's in vivo chemistry. You can do precision changes in complexes and molecules and systems, and that's awesome. The second is it's a beautiful interweaving of concepts and logic. And there are a few things that will make me very angry. But one of them is the Steph Curry haters. The second is people that say genetics is just a tool. And to me, that's sort of equivalent to saying you give somebody a protractor and you show them how to use it, and now you say you're an expert in Euclidean geometry. The third is the intimate tie to biology. And so we see lots of reports in the literature, about beautiful studies where people figure out the molecular mechanisms of some process. But as a geneticist, you kind of always have in the back of your mind, what's the phenotype? What's the value of this whole thing to the organism? And the last thing is the intimidation factor, which is, you tell somebody that you're a geneticist and they think you're smart. And in particular, if you are hanging around with uppity mathematicians, physicists, or chemists who look at lowly biologists, but if you tell them you're a geneticist, there's kind of this Seinfeld moment where they think that you know something about their DNA. And that just gets them a little bit nervous. So that's all great. But the biggest reason to love genetics is the surprises. And what I love most is it proves how stupid I am. Inevitably, it reveals the unanticipated gems of biology that I never anticipated. And I realize that this was sort of a double positive here, but I really think it emphasizes the fact that many times I was totally clueless until genetics told me. And it reminds us that evolution is much smarter and more creative than we are. And I think sometimes we get sort of in our hubris as humans, we forget that CRISPR was invented by nature, not by us. So that brings us down to where do we get the genetic surprises come from? And my colleague Vincent Guacci comes into my office and says, ponder to null or not to null. And of course, the ability to make null alleles at will with recombinant DNA really propelled bacteria and yeast to the status of the model genetic organisms. It's been the envy of everybody. And now that we have CRISPR, this can be done and people are very excited. But there's always a but. And I'd say the but is that many of my lab's biggest insight surprises have come from not null alleles. And this started really in David Botstein's lab, where a not null allele sort of allowed us to determine that proteins could cross the plasma membrane post-translationally, which was a heretical idea at the time. So what I'm going to do today is tell you about insights and surprises in 3 areas of research that we've been doing in the laboratory in the last 15 years: stress biology, genome instability, and higher order chromosome structure. And in the end, I'll make a few comments about how this maybe think about CRISPR. So stress biology through the lens of desiccation tolerance. And what I really want you to sort of think about here is that when I came to the realization that up until really almost the 1990s, all of biology was driven by looking in sort of extreme situations like desiccation tolerance because of a way to get over technical hurdles. And so as an example, we all worked on bacteria and phage because of their huge numbers and speed of replication allowed us to figure out the central dogma. Similarly, people studying tetrahymena, the chromosomes are shattered to 10,000 pieces, allows you to study the ends of chromosomes. So we thought that even though stress biology had been studied for decades, that we might learn something new and unusual by studying it through this extreme condition of desiccation tolerance. And these are the folks that have worked on it. And I want to remind you that in yeast, it's conditional, so that dividing cells, about only 1 in 200,000 cells survive desiccation. But if you nutrient starve them, about 1 in 3 survive. So why don't these cells die? Well, they must have some stress effectors that sort of potentially mitigate the damages of this of desiccation. And so there were in fact established stress effectors for things like protein aggregation, promoting protein stability, homeostasis of osmolarity, oxidation, chromosome, and membrane integrity. We knew about all those. And then also in all desiccation tolerant organisms, there are evolutionarily conserved factors, trihalosis, and these small intrinsically disordered proteins called hydrophilins. So those seem like good candidates. So Dean Callahan did what you did, made a null, looked through all the nulls in yeast, and found that in fact, none of them conferred desiccation sensitivity to yeast, which was kind of stunning. In fact, made us kind of feel like, is this some kind of bizarre, mythical property? But fortunately, Hugo Tapia followed this up and discovered that indeed, if trihalosis and one particular hydrophilin called HSP12, when knocked out individually, don't cause desiccation sensitivity, but together do. So they are together are necessary for desiccation tolerance. Okay, so this was a satisfying result because of course they're evolutionarily conserved and maybe explains why they're in these organisms that are desiccation tolerant. But Hugo then decided to use a not null allele to test the sufficiency of trihalosis and HSP12 for desiccation tolerance. So you start with desiccation sensitive cells. They're low in these factors. And now you engineer them dividing cells, which are normally desiccation sensitive, to have high levels. And then you look to see whether they're sufficient for desiccation tolerance. So did we expect this experiment to work? And the answer is, of course not, because these effectors of HSP12, Hugo had studied quite a bit. And what we knew was they protected against protein aggregation, they promoted protein stability, and they probably promoted membrane integrity. But we didn't have any evidence that they did anything for these kinds of stresses or many other stresses that we couldn't even imagine could happen. So basically, if you think cells die 1,000 deaths because they're desiccated, then you've cured them with 300 and they still have 700 to go. And so of course they're gonna die. So the answer of course was they survived. So here we see the survival of the normal starved cells, 1 in 3. Here's the dividing cells, 1 in 200,000. If we have these cells have high levels of HSP12 or trihalosis, we now survive 1 in 100, which is a lot better than before, but really impressively, if we have them have both of these factors, now we take these dividing sensitive cells and drive it up to 8 in 10 cells survive. So they are both necessary and sufficient to promote this amazing trait. So what does this thing tell us about stress biology? Well, first of all, despite 50 years of studying stress biology, we've now identified two major new classes of very powerful stress effectors. And we believe we have evidence that they aren't, their function isn't relegated simply to when desiccation occurs. Furthermore, the hydrophilins, so there's 11 of them in yeast, this is just one, and we think they're involved in many other interesting aspects of membrane biology that we have yet to tap. The second thing I just want to focus on is what kind of model we have for why they're sufficient for desiccation tolerance. And the way we think about it is that there are a few stresses that desiccation enhances, but they are mitigated by HSP12 and trehalase. So these things are dedicated molecules that can deal with a subset of stresses. And then we sort of flip the rest, is that maybe desiccation really isn't as stressful as we think, that desiccation actually eliminates most stresses because they are potentiated by water. So actually removing water makes a less stressful life. And I want to just throw out one little fact, which is something that's sort of a little bit unrelated to this, which is that we've discovered that in the desiccation stage, there is some metabolism going on. And so I think together all these observations have an implication for possibly the origins of life, which I'm happy to talk about in the discussion. So the second area of research I want to tell you about is genome instability through the lens of R loops. So we're very interested in how chromosomes are properly transmitted, but also what can go wrong. And R loops are where the RNA transcript hybridizes back to regions of homology on the DNA, generating this structure called an R loop. And really was, at least in eukaryotes, was pioneered by the work of Andres Aguilera, who kind of worked solo on this for a while until my lab and a few others discovered, these are really cool. And now it's a huge cottage industry of a lot of exciting observations. And so the R loops can both not only cause genome instability, but also regulate gene expression. So they're quite important in biology. And the type of instability is these gross chromosomal rearrangements. And the presence of R loops has been correlated with cancer and neurological disorders. And this is the team of folks who worked on this over the years. So how do R loops cause genome instability? Well, this unusual structure is subject to induces a double strand break. And what people have thought and known actually is that once you have a double strand break, those kinds of lesions are actually prone to induce genome instability and death. So Jeremy Amon wanted to look at this in a little more detail and to understand what causes sort of the factors involved in the genome instability. And he looked for suppressors of genome instability and associated death. And the idea is, well, what would you expect for such a screen? You expect the outcome that you get mutations that block R loop formation so that you never get the R loop forming and therefore you don't go this pathway and eventually get genome instability. Or you would get mutations that improve the repair pathway such that you could repair these lesions and go back to forming a normal chromosome. And of course, in the theme of this talk, that's not what we got. Jeremy got a mutation. Here's the normal cells, which are are very sensitive and die because of persistent R loops that cause the cells to die. And Jeremy got an allele in this PIF1 gene, a not null allele, which brought them back to life, preventing somehow the lethality associated with high hybrids. And it turns out that what this PIF1 allele does is reduce the activity of this protein. And what we knew from the literature is that PIF1 protein is actually required for recombination-dependent repair. So this turns the whole argument on the head. It seems as if that it's not that what's killing these cells is a misguided attempt to repair them. And if you block the damage associated with R loops, then actually they survive. So Lorenzo Costantina sort of took this on and inspired by this and other reasons, developed an assay which really for the first time could look at the lesions and the repair associated with those lesions as a consequence of R loops in a very ingenious assay. And what he discovered is that when the cells have damage due to R loops, they go into a panic mode. And they do some very bizarre things. They take one end of the R loop and they insert it into a neighboring a homologous piece of DNA and proceed to do, one-ended DNA replication for kilobases, tens of kilobases. Alternatively, they can take the other end of this, other end here, and resect the DNA, making huge regions of single-stranded DNA. So these are kinds of lesions I think has never been seen with any other kind of DNA damaging agent. And so in the end, we have a big mess, but we think it's a really interesting mess. And in summary here, what this kind of surprise we got from this not null allele is that the field really felt that the reason that R loops caused genome instability and possibly cancer was because they increased the double-strand breaks, which we knew in a sense had some propensity to do this on their own. But what this study really says is maybe the reason is not that they're actually generating so many more double-strand breaks, but it's the type of double-strand breaks they're generating, which is in fact pushing these cells into this very unusual process of DNA repair. And so this, observations, I think, explain the possibilities of why you get cancer and, DNA-associated large rearrangements with R loops. Okay, so the last element or topic I want to talk about is higher order chromatin organization, which has been sort of one of the major focuses of my lab, not only the last 15 years, but before that. And these are the people that have been working on it in the last 15 years. So the story begins with the identification of really a not null allele by Alex Strunikoff, which affected plasmid segregation in yeast. When he cloned that gene, it turned out he discovered to his great excitement that it was a conserved family of proteins that was present in humans. But to his great dismay, it was actually also present in bacteria. So he was hoping to identify a kinetochore protein and this sort of eliminated that possibility. But I was excited because of my roots in bacterial work. And I realized that something that's conserved from humans to bacteria is probably a protein that organizes chromosomes in all types of life. After that, very quickly thereafter that, these subunits were found as part of complexes in the cohesin complex, which Naismith in my laboratory identified, and in condensin-like laboratories, which was identified Tatsuya Rana and Barbara Meyer's laboratory. And the idea about how these things work is that they tether together DNA strands in cis and trans, or in cis. And that's the way they could modulate chromosome structure. So how do they do that? Well, there was a very simple model that was put out there, an elegant model really by the Naismith laboratory called the Embrace model, which took advantage of the fact that in the electron microscope, what one can see is that a fraction of these SMC complexes are present as very, very large rings. And so the idea was you could entrap or tether decays to DNA by entrapping them within the ring. And over the last 15 years, we had gotten a bunch of not null phenotypes that were inconsistent with a simple Embrace model. First of all, we found that there were multiple distinct DNA binding activities. These alleles told us that that the complex likely went under large conformational changes because we could see genetic interactions between the head and the hinge domain. And what we found was that the ATPase alleles of that in their biochemistry, study of their biochemistry, they remained active. And the thought was that the ATPase was required for DNA binding. So why would you keep the ATPase active after your DNA binding? And finally, we had genetic interactions, which is said that cohesins act as multimers. And I point out to a really beautiful paper that was just published by Sihang, which actually takes that genetics and provides now new molecular assays for cohesin oligomerization and understanding its biological function. So these observations were whispering to us that this model was way too simple, but really they were shouting at us and we weren't listening. Unfortunately, the really big surprise came from the biochemists, which is really hurts to admit. It's kind of like admitting that Stanford discovered something before Berkeley. But in some really beautiful experiments by Christian Herring and Seth Decker, and then really some beautiful in vivo experiments of bacteria by the Rudner laboratory, what was discovered was that the SMC complexes are actually DNA translocases, which use the ATPase to extrude loops of DNA. So you can see these loops by this really cool assay that was developed by the Joe Decker lab called chromosome conformation capture. And important for this assay, all you really have to know is that when the two stems of a loop get together, they will appear as a spot off the diagonal. These are chromosome coordinates here and here. And what you can see in mammalian cells is you can see spots all along the diagonal indicating that positioned loops like this are a prominent feature throughout the genome. So we got into this, and well, I should say before we got into it, there have been attempts to look at loops in budding yeast, and you could see them occasionally, but they didn't seem nearly as prevalent or widespread as they were in other organisms. So we did get into this point, and we this time is Lorenzo Costantina, Stanley Zai, who's a member of the Darzak lab, and Rebecca Lamoth, a really talented graduate student. And there are many, many cool observations from this paper, conclusions, but I just want to focus on one, which is now with an improved method that Stanley had developed. What we could see was that yeast were really no different from mammalian cells. There were positional loops along the entire length of chromosomes throughout the yeast genome. So now the function of cohesion has gotten more complicated. We knew they were DNA tethers that held the chromosomes together and do things like sister chromosome cohesion, DNA repair. But now they did this loop extrusion. And at the time, the idea was, because they were discovering mammalian cells, first that they were a way of controlling proper transcriptional regulation by controlling the interaction of promoters with their enhancers. And that idea got a little bit muted with the discovery that they actually probably only controlled a limited subset of genes. And this didn't really account for why then they were prevalent throughout the genome. And now we find them in yeast throughout the genomes, and yeast doesn't have this sort of enhancer promoter problem. So that led to the idea then that maybe they're doing some evolution conserved and more important function than transcription. And I put that in there because I like to tweak my transcription friends. Okay, so the question is really what the heck are these cohesion loops doing, and how are they formed, and how are they regulated? And so this is the current team that's working on that. We're just beginning because of the pandemic. We haven't gotten as far as we'd like. But I want to tell you one little tidbit of new data, which is sort of fits with the theme of this talk. And it really involves Vinny Guacci. Vinny, as you can tell from this picture, is not a null human. But he has been, he's just a live wire with full surprises. And more importantly for me, he's been my sort of collaborator and co-leader of the lab over the many years, and has, without many of our key observations and discoveries, would not have been made. So Vinny is looking at, is the regulation of cohesion looping important for the fidelity of DNA repair? And so normal DNA repair happens where if you have a break in the DNA, what happens is you repair off the replicated molecule, which is held nearby, restoring the normal copies, and those get segregated to the daughter cells. But if you have repetitive sequences in the regions that are also homologous to where this damage occurred, the cells can use these DNA distal as a template for repair if it forms a loop. And if that repair reaction happens, and this is sort of making even you hardcore geneticists go back and put your Licton and Haber brains on and follow strands and all that kind of stuff that makes your eyes cross. But in the end, the repair reaction can generate this unequal cisterone exchange events where you lose information intervening between the two repeated regions, and you duplicate the genome. In addition, in this particular example here, what we have is the repeated region is a gene which has its C terminus removed on one repeat and its N terminus removed on the other repeat. And the recombination reaction restores the full copy of the gene. In this case, it's the UR3 gene, and now these yeast cells can go on uracil. So we can easily detect these unusual events by this new phenotype. And it's important to think that, of course, the amount of this recombination with these repeats will depend maybe upon how far apart they are from each other. So the bigger they are, the bigger the loop you have to make to bring them together. So Vinny did this experiment now with repeats that are 3 kb and 68 kb apart, and we looked just in wild type cells. And we can see that there are rare events. They happen about 5 in a million, 5 times on 5 times, yeah, one, whatever, it's 5 times 10 to the 1, that should be 10 to the, yeah, 5 times 10 to the minus 6 and for closed loops, and they happen even more infrequently in cells with where the sequence is far apart, indicating that it's harder to make larger loops. And in fact, Lorenzo and the crew had seen this with their chromosome capture data. So now what we can do is we can use a WAPA mutant, which increases the frequency of large loops by increasing the persistence time of cohesion on chromosomes. And as we might expect, now these cells can make bigger loops more easily. And lo and behold, now there's no difference between having interactions with the close by repeat or the repeat that's very far away. So this was all great, fit beautifully with how we were thinking about what loops did. Then Vinny turned to a not null allele, an allele in the SMC3 subunit of cohesin, which reduces the ATPase in a beautiful study that he and colleagues had done, Kamdari. And so what we think is because it has a reduced ATPase activity that the complex gets on, but it can only make much smaller loops because it doesn't really have the engine to drive it to make large loops. So we expect from this particular kind of mutation, inability to make the large loops, so that interactions with the distal sequences to unequal sister chromatin exchange should go down. And lo and behold, what we found was it wasn't the case. So a surprise, which is that this so-called mutant now actually also increases the ability to interact with sequences distal to the loop. Okay, so I think it's just a tidbit of the kind of surprises we're going to get as we put our battery of not null alleles, which allow us to precisely change aspects of this protein complex function, to what it's going to do to the mechanism and biology of this organism. But let me just start by saying, what have we learned? Well, we've learned that the proper regulation of cohesin function is required for the accuracy of sister chromatin exchange. It had already been known for years from work from Hartwell and also the Shorgan and Naismith publication that you prefer to use the sister as a template, but we're taking it one step further and saying, actually, it's not just you use the replicated molecule as a template, but it helps to determines which of the sequences on the replicated molecule that you use. And this can be important because the idea of copy number variation is clearly a huge feature of normal cells, and this may play a role in that, and explains the fact that cohesin mutations are amongst the most prevalent in cancer, and may explain the copy number variation and deletions in cancer, which are thought to cause tumorigenesis. And so the biological function of cohesin loop extrusion, however, is a little bit on hold because we didn't get what we predicted from all this. And so I stand by for the further analysis, I think, which is going to be exciting with what we're trying to do. Okay, so I'm going to end now with just a few thoughts on CRISPR. So just remind you, this was my journey to genetics. And I want to just point out Jennifer Doudna's. So she was a biochemistry major, sort of like me in college, but wasn't converted to genetics until really 2012 when she published her paper on CRISPR. And since then, she's won a few awards. And I just would like for all of you to ponder the concept that what would have happened if she had met David Botstein back here and been converted to genetics much earlier? Then she could have discovered something really important. Okay, so of course, CRISPR is revolutionizing biology. One science, we can see this in headlines everywhere. People are incredibly excited about it, scientists and lay people. There are a few naysayers, the but people, and we can take that with a grain of salt. But I think there is a but. And I think the real but is that CRISPR gene editing currently surveys only a small portion of biological space. And I guess my history in thinking about yeast is we have to be aware of the seduction of CRISPR editing because of the ease of making nulls as a way to identify genes and gene interactions. And of course, that's powerful, but I would say that the yeast field has sort of been stuck in this rut for a long time. And that I want to point out that I hope you appreciate that most of the non-null alleles described in my lecture, none of them could currently be identified by CRISPR because they involve unbiased genomic genes, changes to sort of single base pairs. So in this case, CRISPR can't come up with any of the insights that we've obtained. which, so I want to end the talk with the following idea, which is simply that, of course, you want to make nulls and study there. That's clear, and you want to exploit this property of CRISPR. So you can join the herd and you can read the thousands of papers where they're doing this kind of approach. But I hope that some of you might venture into the not null space and enjoy the solitude and the surprises. So I'm going to end by thanking the lab for and the GSA for this chance to give you a talk. So I'll stop there.
Thanks very much, Doug. That was quite a exploration through a wide areas, and yeah, I'm a big fan of non-null alleles as well.
Yeah, I think I'm into the choir here, but anyway.
We had a couple of questions coming in. I encourage everybody to put your questions in the chat. If you'd like to just ask your question, let me know and in the chat, and I'll just call on you. But Tom Pettis was wondering, actually, do you want to ask this or do you want me to read it, Tom? I guess I'll go ahead and read it for now. Does your data on the types of recombination events caused by R loops argue that R loops are not likely to be associated with most spontaneous recombinogenic lesions?
Yes, I think that would possibly be true. I think that, I mean, we don't know because now we have other questions, because the problem with geneticists, as I said, is they are smart. And so they're going to start asking the tough questions. The answer to that question is, in order to be able to see those events, we use a sensitized system. And so one always has to worry a little bit that some of the results you got was a sensitized system, that maybe in the totally normal background, some of the events might have been repaired by a normal process, but I don't think so. I think the most likely term is yes.
Thanks.
And sorry, did you have a follow-up question?
No.
No, okay, great. So Barbara Meyer is commenting that we find tons of non-null alleles with CRISPR mutagenesis.
Okay, well, that's it. Barbara's always going to smack me, but I guess has she done unbiased genetic screen? Of course, we make non-null alleles all the time, and we can make them to some extent. I guess if I had to look for suppressors where I was expecting to get amino acid substitutions, specific amino substitutions, I might not, in an unbiased way, you wouldn't use CRISPR. But Barbara's always ahead of the curve on me. That's why she's the master and I'm still the student.
Doug, of course, you get a variety of mutations, and depends on how you do the mutagenesis. What if you have repair templates? Not, depends on all sorts of things. So you can skew it to go whichever direction you want. You can use it.
But the repair templates. So this is why Barbara and I love. We teach class together, and we get in these kind of fun arguments all the time. So, but Barbara, then you can't put, if for example, we were looking for suppressors that the Vinnie's allele, the way we got that suppressor I talked about at the end was we were looking for things that would bypass the need for cohesin to be acetylated for its regulation. And that was a surprise in itself, because we got a change in the conserved, absolute aspartate is conserved in every single organism throughout, from bacteria to humans, and it was a change to a glutamate. Now, nobody in his right mind would ever make that change, and I don't think you could get that by CRISPR.
Right.
You wouldn't be. You have to make a million templates and change everything, and you would never design that change, because who in his right mind would ever make that substitution?
Yeah, I'm talking about non-templated CRISPR mutagenesis. You get a huge variety of things there. You're absolutely right about the mutations we would not have gotten, but I'm just stressing the other option, that you can use it wisely and get many surprises that are extremely useful. That's all my points.
Cool. I submit. Okay, which is usually the case whenever I get in discussion with Barbara. But anyway, what else have we got here?
So Sue Jinx Robertson is asking, you're seeing BIR with an R loop, correct? So R loops are primarily associated with one-ended events, whereas normal homologous recombination events are two-ended.
Yeah, that's right. And we think that's because the R loop, actually, the persistence of the RNA stuck to the end of the DNA is actually what keeps them from being able to do the two-ended reaction. You're basically chemically blocking the one end from being recombinogenic. So that's what happens. So I don't see any more questions. So I'm actually going to do my origins of life pontification, which is what happens when you get old and you have white hair.
That was indeed what's going to be my next question for you, was to take us down that origin.
When I listen to Jack Shostak, who's another master, the idea is that you start in this, the real problem is, to get some of the chemistry, like making an RNA and then duplicating an RNA, and that happens in an aqueous environment. And so that's the problem they're trying to solve. And then the question is, but of course, in nature, you have to concentrate that chemistry somehow. So, you have to get a membrane around it. And that seems to me is a really hard thing to figure out how you do that. How do you compartmentalize this stuff to make it work? So maybe what we've learned from desiccation is that, in fact, and it's a stressful environment with lots of things happening. So maybe what happens is you actually start in an environment with almost no water, like an oil droplet. And you have just a little bit of water goes in, which allows you to do chemistry, but that prevents you from a lot of the stress that you would normally have. And then over time, what evolved was systems which allowed more and more of the water to get into this thing more actively. And so that's the way I think sort of maybe the evolution of life happened. Of course, that has a problem, which is, how do you get an RNA molecule into, a highly charged molecule into this environment? It's sort of, I'm solving one half of the equation and making the other one harder, and he solved one easier and made the other one harder. But it's just an alternative idea. It's certainly something to think about for those folks that are looking for life on different planets, is that metabolism can happen in the absence of water, and water is maybe good and bad. It generates a lot of stress.
So it looks like a comment to that has come in from Tom Klein, saying that Nick Lane discusses naturally occurring small compartments that could have served membrane function early on.
Yeah, I guess the idea is that there are little niches in rocks and stuff like that they could have begun to sort of initially concentrate it, and something could have happened otherwise. That's certainly, I think, one idea I've heard. I did write Jack with this idea because this talk kind of inspired me to think about it, and I haven't heard back from him yet, so it probably means it's pretty lame. But anyway, I'm hoping I'll tweak him a little bit, see what happens.
Could you tell me, are the SMCs among the 500 totally conserved proteins that are present in all, archaea and eubacteria and so forth, or are they a later evolutionary, even though they are in bacteria?
Yeah, I don't know that as well as I should. I think there are some bizarre organisms that don't have them, but there are. It's not absolutely. I don't know how well the genomes of those are known, but so those would, I could argue, were the ones that lost it more likely than had it. But as far as I know, they're present in everything. But so absolutely conserved is, certainly means that they're present. The family is present in all. The proteins themselves are pretty divergent, but in terms of their amino acid sequence, yeah. So,
hey Doug,
it points to the kind. And I should say that, I think one of the most beautiful studies that really is sort of underevaluated and appreciated is David Rundner's laboratories, which showed really the first time that large loops, these complexes were capable of making very large loops, which, so far is that it wasn't, an in vitro artifact that they could make large loops, not that they don't make loops. The problem is we don't really, so that means they're very anciently conserved, right? They're doing something, and I don't think it has to do with enhancer promoters, and whatever that activity is, it's clearly being used in yeast and humans. So it likens to there's some really ancient activity that they're for. The interesting thing is I don't think we really know what they do in bacteria. They have a pretty mild phenotype, which is shocking, considering how complicated, complex they are, which again makes me think that there's some really big aspect of the biology that we don't get.
Hey, Doug, I have a question about the R looping.
Yeah.
So we know in bigger eukaryotes, a lot of the regulation of expression that happens with releasing paused RNA polymerases. They've already initiated, so they've already made at least a little product. So how big does the hybridized RNA have to be to cause this instability? Because you'd think if there are a lot of paused RNA polymerases that have made at least some RNA DNA hybrid, there'd be a lot of instability in a mammalian genome.
Yeah, so I mean, I think the answer is we don't really. I think the general size we think is 400 base pairs, which is much bigger than usually the amount of paused RNA that's there. And I think, there are complexes, it may depend. I mean, what is it about this structure? Is it that you have a large single-stranded piece of DNA that makes it originally a part of the target? We don't know. I should point out that, one of the contributions that Lamia Waba made in my laboratory was to show that almost any perturbation to RNA biogenesis leads to the formation of R loops, that they can actually be formed by splicing mutants, things that don't export it from the nucleus, things that reduce turnover, all these kinds of things, which is why they're quite prone to happen.
Hey, Doug, Scott.
Yeah.
What determines the spacing of the SMCs in making this array of loops?
Okay, so that's a good question. I forgot to say that, I was going to put somewhere in my talk that we worked on many, many different things in my career, including one foray into vacuoles with Scott Emmer, which was an amazing experience. But anyway, so the answer is that, so that's one of the things I think that's been really cool about the in vitro work, which I have to say I was a naysayer about this whole phenomenon. Part of the reason I wasn't listening to those little cues that our mutants were telling us about was that what we knew from the biochemistry was these were really crappy ATPases, and anything that translocates along DNA, any of the motors, whether it's a helicase, they're the most voracious ATPases that exist. The other part that made no sense to me was that they were supposed to be marching down chromosomes, 50 to hundreds of kilobases in length. And I just couldn't believe that they would do that like as a sort of typical motor and remove all the nucleosomes. Because that was just totally disruptive to everything we know about silencing and control of gene expression. And so how could you possibly have something motoring down DNA like this? Well, what the beautiful experiments from Decker and colleagues showed was that they don't move in nanometers, they move in 50 nanometers, which allows them to step over nucleosomes. And so the idea to think about them is they're like those, the big creatures in the Star Wars or whatever, those things that the guys were driving at the end to fight the what are the Ewoks, which is, they're like these big things and they move along in these big steps. And that's part of the reason possibly, that the Koshland complex with the Genome are huge. They're incredibly long. They're 50 nanometers in length, which is, bigger than 2 ribosomes, even though they're a little complex. So it gives them the ability to have very long legs to move. And so the answer is, we think they can move 50 nanometers. We think they could step over nucleosomes, but I wouldn't put that in a textbook. And the big problem for those in the field is how do you get directionality? So in most cases, when motors move along something, they're moving along an asymmetric template. So it's easy for skeletal motors like actins and microtubules. It's a problem for DNA unless you open up the DNA, and then you can move along from 5' to 3' or 3' to 5'. But a double-stranded piece of DNA is a symmetric substrate. And so you can make sort of ways of trying to make reactions to these things, but it's a really big, I think that's the biggest challenge in the field right now is how to make these things directional.
Maybe that's where double-strand breaks come into the story.
Yeah, that certainly can provide some way to make it, but they certainly move on pieces of DNA that aren't broken, we think. But maybe you're right. Maybe there's a nick or something generated, or some kind of structure of the DNA that's changed. That's not a possibility.
So Susan Gerby has a hypothesis here. So when a replication fork stalls and is prone to breaks, the RNA primer on the leading nascent strand could be likened to a mini R loop. She's asking if you can feed that into your model, and said, great talk.
Yeah. Yeah, I mean, it certainly is possible. I mean, I guess what I'm trying to think here is, yeah, so the question maybe why stalled forks are particularly mutagenic. It mightn't possibly because they also invoke an R loop intermediate. That's certainly a possibility. Yeah, I think that's, and I don't know. I mean, we certainly, we could try and localize. I don't know if anybody's done the experiment of asking whether R loops localize to stalled forks. It would be an interesting experiment.
There's a question here from Giovanni Bosco asking, what do you think is the ancestral function of SMCs, cohesion or condensation? Or are these two general functions more similar than we think?
Yeah, that's really interesting. Well, I actually think it could be DNA repair, which could be the most ancient function, possibly. But, you know, I mean, they could be related. So certainly from the bacterial work from Rudner, what you see is that in this particular case, the motor activity pushes the two newly replicated strands of DNA apart. And so, you know, in that case, it's sort of, in some ways, acting as a segregation function, and you know, might be equivalent to condensation. So I think the motor activity was the most ancestral, and the way I think about it is that that the tethering activity is just a stalled motor. It's like a myosin in a Riger state. That's, I mean, once you argue that there are multiple DNA binding activities, which is what you would need to be able to move along something, all you have to do to make it a tether is to trap the intermediate in the reaction, stop the cycle, and now you've generated a tether. And that actually is generating all kinds of, I think, thoughts that people haven't really put into mind, because there is very distinct, the idea that the most recent experiments from Jan Michael Peters' lab, which says that in order to act as a motor, you never have to put DNA inside the big ring. And yet the embrace model and even us have considered ideas that the tethering happens by putting DNA in the ring. And I don't think that's possible. I don't think that you invent multiple different DNA binding activities. That if you believe the idea that it's just a stalled cycle, tethering is a stalled part of the cycle, then we've gotten something quite wrong in the past in terms of how we've thought about how these things are tethers. So it's challenging.
Great. So are there any other questions?
Well, thank you all for coming. And as I said, go out and drink a beer and think about how great it is that you have a wonderful group of a field of people and good colleagues, and NIH likes you.
Thanks very much.
Congratulations, Doug.
Congratulations, indeed.
Good job.
Congratulations.